Okay, hello, my name is Nychelle Bowles. I am from Trinidad, Colorado, and I go to school at Colorado School of Mines. My project this summer was separating silicon quantum dots using thermal field flow fractionation. I am using thermal field flow fractionation to separate quantum dots because it has a good sample loss recovery, and also because it's kind of cool. Thermal field flow fractionation uses a temperature gradient along with the regular flow field to separate out small particles. What happens is we have a regular diffusion which causes particles to move up towards the higher wall of the channel, which is called the hot wall because it's heated, and we'll have a thermal diffusion which causes particles to move closer to the cold wall. This causes a size separation kind of base particles because particles that are smaller have a higher D, and they tend to go towards the middle of the channel where the flow is a little bit higher. So we end up with smaller particles eluting from the channel first. The silicon quantum dots that I am attempting to separate are passivated so that they basically attach a 12 carbon alkyl group to the surface of the silicon quantum dot. This helps prevent oxidation and preserves the properties of the silicon quantum dot. The way that they make these quantum dots does not ensure that they have a narrow size distribution. So my project is to separate these into smaller size distributions. The first experiment that I did was to determine if the temperature gradient on the thermal field flow fractionation caused any problems with the silicon quantum dots. What I did is I varied the temperature gradient from 0 to 100 degrees C and took hydrodynamic radius measurements using quasi-elastic light scattering. And basically what this told me is that no thermal field flow fractionation does not affect silicon quantum dots, and that we are actually able to see silicon quantum dots in the main size range of the size distribution determined by transmission electron microscopy. The second part of my experiment was to determine which reactants would contribute to what in the fractogram. So I just ran the carrier fluid and I got a rather flat line, which is very good because we don't want anything strange coming out in our carrier solution. And then I ran unreacted dodecine and reacted dodecine to see what those two fractograms look like. And then I ran silicon quantum dots. What I could basically tell from this is that this 1 dodecine fractogram is actually contributing to the silicon quantum dot fractogram. This is just good to know because I want to make sure that we are actually not just picking up on silicon quantum dots, but we are picking up on the alkene that is used to passivate the silicon quantum dots. The third part of the experiment was to collect fractions. So I used a delta T of 100 degrees C, and I collected 3 different fractions. I collected the first half of this peak here, the second half of this peak here, and the last and final, this small peak that comes out separate of the thing. And then I took TEM images of them to determine what's actually coming out of them. The first TEM of fraction 1 indicated that I was getting particle sizes from 2.5 to 4 nanometers, which is pretty good narrow size distribution there. And then the second TEM, I was getting particle sizes from 4 to 6 nanometers, again good size distribution. And this final fraction here, I got a lot of aggregates, and a lot of aggregates that are spherical in shape from 40 to 60 nanometers in diameter. This indicates that I'm losing all the aggregates in one peak, which is very good because we can't really use those, so it's nice to get rid of them all in one peak. Further steps to go with this project is one, narrow the amount of the peak size that I'm collecting. So I'm going to just collect 1/3 of this and then another third of this and collect that first peak until 3 parts. That way we can see if we're actually getting even narrower size distributions than what is first indicated in here. And possibly using other methods such as size exclusion chromatography or possibly even flow field fractionation to determine if I can actually get rid of that 1 dodecine, which is useless to everybody, and so I just have silicon quantum dots in a purified form. So that's it.